From Microbe TV, this is TWiEVO, This Week in Evolution, episode number 75, recorded on February 21st, 2022. I'm Vincent Racaniello, and you're listening to the podcast on the biology behind what makes us tick. Joining me today from Salt Lake City, Nels LD.
Hey, Vincent. Good to be with you from snow-capped LD Lab Studios.
Snow-capped, huh?
Yeah, I've got finally a little bit of snow here. We've been, winter's been a little dry. We started out great in December, and then things kind of went off the rails here, really dried out, but we've finally got a few systems coming through, and I'm happy to report that there's light snow coming down from here. How about there in New York? What's the current conditions?
We had snow a few weeks ago, but there's no snow here in the city on the ground. It's rather cold. It's been on and off, cold and warm the last few weeks. It's been weird. It's right now 10 degrees Celsius, but no snow. Have you been skiing this year, Nels?
Well, just a tiny bit. So I've been, a little, well, there hasn't been much snow. There's a good base, but we're always, we're kind of snow snobs out here looking for that fresh powder.
Yeah.
And that's been a little bit limited. So I've been sitting it out for a little while, but I'll get back in for sure here in the next couple of weeks.
Before the season's over. Yeah.
Yeah, exactly. Yep.
So what's a COVID situation out there?
Yeah. So COVID update, things are cooling down. We're definitely coming down from the peak. I think a couple of weeks, 4 weeks, maybe behind. I'm guessing where you are in New York, but moving in a better direction. At the same time, I still it feels almost surreal to me how, quickly everyone is ready to sort of move on. And while we're still, the cases are down, but the trailing, hospitalizations and deaths are still remarkably bad, just in a relative sense. And so, and then of course, thinking about this a little bit from an evolutionary standpoint, I think there's a lot of evolutionary virologists out there, genomic epidemiologists, sort of holding their breath. Is there another variant in the wings? And I think the answer as of today is we don't know.
Who would know, right?
Yeah, exactly. But that sort of, process, that evolution could have been playing out even months ago, and just sort of sitting, waiting for conditions like this, right? So I think Omicron's been a remarkable sweep across the globe in, less than 6 months. And at the same time, we've tracked this like we've never tracked a virus before, but it's still, I think it's caught a lot of people off guard how quickly that could move.
Well, as you say, we're tracking. I'll bet this has happened before. We just didn't see it because we weren't looking for it, right?
Oh, yeah. And, as the evidence has come out, the sequencing has come out on Omicron and even the variants there. So the BA.1, the BA.2, which is now sort of filling in for the first surge, even as cases come down, thankfully.
Mm-hmm.
There's, that didn't just show up at our doorstep in, say, December. It was percolating back last summer, maybe even a little bit before. And so that raises the obvious question: is there other variation out there now? And then, so it's in a sense, I think it's like a new challenge or a new obstacle or test for SARS-2, which is now there's a lot of pretty fresh immune memory of what SARS-2 looks like from the massive sweep of this. And so that I think, that means that, hopefully there's, I think what the epidemiologists call a little bit of a honeymoon now, where the R value goes below 1 because of how fast the sweep went. But then, what's waiting there in the wings? And as we certainly know, if viruses do one thing, it's they adapt, they evolve, they are constantly invisibly testing the immune defenses in populations. And so if something's possible, I think it's probably a matter of time before we're back into another variant.
Probably the masters of evolution, Nels, I would venture to say. Viruses.
Right.
So let me shout out to some of our I'm amazed everyone came back. We haven't done a TWiEVO live in a while, and I just put it up this morning, and everyone's here. We have MK from sunny eastern Massachusetts. where it's 51F. I know that Andrew is in New Zealand, for example. Mr. Keidman is in Sweden, where it's evening, snow blizzard. Wow, that's pretty cool. And Environmental Coffee House, who's here in New York State, calls it weather whiplash. That's a good name for it, right?
Yeah.
And
A
look
little
at
bit of that.
Della
Go
Sandra
ahead.
says that TWiEVO is the best. That's cool.
Hey, good to see you. Thanks everyone for showing up on short notice. And kind of, I have to say, Vincent, kind of a fun landmark here for us. TWiEVO 75, I guess the diamond anniversary of our of our podcast. If we're on our current pace, I think that puts us at TWiEVO 100 about 2 years from now. And so, maybe we should get a little organized and start booking our rooms, and TWiEVO goes to Hawaii or something like this. I don't know.
Or you could come to the incubator, Nels.
Yeah, well, let's do that for sure. Hopefully before 2 years. I would love to get out there and see the new setup. Yeah.
Okay, so Andrew has the trick. Set the notification bell to all. I don't have any notifications on my subs, so maybe that's it. And Della Sandra says, but Vincent doesn't really trust the R value. So it's not a matter of trust. Here's the thing.
Yeah.
I mean, what is the R value? The reproductive index, right? It's a measure of how well the growth of the viruses in the population. I hate to say transmission, because if you can bring the R below 0, as Nell just said, you can't be affecting intrinsic transmissibility of the virus. What you're doing is affecting population activities, right?
Agreed.
So I do not trust the R value as a measure of intrinsic virus transmissibility. That's very difficult to come by. And Nels, by the way, the CDC just put out a report saying that alpha is no more intrinsically transmissible than ancestral viruses. Took that long to figure it out. And, this is what we have been saying all along. Just be careful. Don't use population observations to infer about intrinsic transmissibility.
Yeah.
Okay.
Oh, yeah. No, and I probably, laid the trap there by starting to try to talk like an epidemiologist. We'll stay on the evolutionary principles today. And that brings us to our paper that we'll cover. Maybe why don't we jump right in here? So
can I put in one question? This is relevant.
Oh,
before
of course.
We start.
Yeah,
Here we
please,
go.
of course.
This is a good question. Bria writes, since Spanish flu ended, how come there were no variants? And we're seeing variants now. And I would say, Nels, there probably were. We just didn't know. We didn't look for them, right?
Oh, absolutely. Well, and also, so the pandemic flu ended, but flu hasn't gone away since. It's endemic, became seasonal, sort of throwing around more, probably too loosely throwing around some epidemiologic terms. But yeah, so that terribly severe pandemic strain, the 1918, 1919 influenza, of course, echoes and ripples that I think continue today, even as we in a bit different beast too. So maybe that gets closer to your question, Priya, is there the way that the influenza genome is organized into the segments, and with reassortment. So the H number, N number flus that show up each year, and then also that very clear moving between animal reservoirs, as we sort of colloquially call this, the bird flu or the swine flu. And so, I think some similar evolutionary sort of things, but with a very different rule book in how the virus is organized and how it adapts and evolves. And honestly, I think we're still kind of playing catch up on the coronaviruses, given how before SARS-2 arrived and everyone became sort of a coronavirologist in one way or another, for probably more worse than better in some cases. But so we'll be talking about this this afternoon, which is looking at some of these virus genomes as they're emerging, or finding new ones that are related to SARS-2, and trying to start to learn a little bit about the rules of how these things change, and with the hope that this might help us both understand what might be out there, and how things might bubble up in the future. whether that's sort of on the spectrum of new variants coming up. I just saw this morning, I think, a review that's suggesting if you look at the phylogenetics of Omicron and some of the immune responses, is this already kind of serotype 2 of SARS-2, where the other ones are serotype 1? Are we starting to come to that somewhat arbitrary, but that definition line of the difference between a variant and a strain? Yeah. What do you think about that, Vincent?
Well, this is something we think about a lot. Amy has been doing a lot of work on serology among enteroviruses, and so the question is, what is a serotype? And traditionally defined as, at least for polio, protection against disease in an animal model, but it's not absolute for sure. And I think Omicron begs the question: is that a new serotype? Yeah, I just was looking at that opinion piece today, and it is. For polio, which really was among the first to have a serotype designation, they said, okay, if you infect a monkey with this polio virus and then you infect with another one, it's not protected. But if you infect it with the same one, that defined the serotype. And so Omicron, reactivity with sera from convalescent or vaccinated people is very low, right?
Yeah.
And that's why everyone can be infected, even though you don't go on to develop serious disease, which is probably because of T cells that whose epitopes have not been changed. But I think, I think Omicron is most likely a serotype. And so how do we do that? There has to be a committee that looks at the data and decides, right? And I'm not on that committee.
Yeah. Yep. No, I hear you. So yeah, and then that's I think we're in this sort of moment of like, maybe catching our breath a little bit, and in some good ways, we're ready to move past Omicron, given that remarkable sweep.
Yeah.
But what comes next? Is there serotype 3, or do things really dive down that's virus forecasting, much harder than weather forecasting, which already is a high degree of difficulty.
Well, for influenza, we don't say serotypes, right? We have we have subtypes, and they're defined by the H and the N, right? But then you can have a lot of different H1N1s over the years. Yeah. You can have annual variants, and over the years they can. And in fact, the 1918 influenza, when it went into people in 1918, it also went into pigs, and it has been circulating in pigs since then. And in pigs, it undergoes much less antigenic variation. And so that is why in 2009, H1N1 reemerged from, in part, pigs, because it was antigenic. Nobody around had lived in 1918. So a version of that virus, not too different, could emerge out of pigs into people.
Yeah. Yep. Yeah. That immune memory was lost with given enough time, which sort of illustrates this interesting tempo between the host population and the viruses, especially as they might be hanging out in reservoir species and animal species as well. So, of course, in an evolution podcast, we're always to try to think about the future. We're always looking backwards. And so I think this paper does that today, although looking at the very recent past. And so the title of our paper, which was officially published last week, is Bat Coronaviruses Related to SARS-CoV-2 in Infectiousness for Infectious for Human Cells. Sorry, I kind of stumbled over the title there. And this is a group of authors from the Pasteur Institute in Paris, from the University of Laos, and also the Pasteur in Laos, a group of virologists and field biologists actually going out sampling, collecting viruses. So the paper was just published in Nature, or posted, I guess, on Nature last week. You can see that's a accelerated preprint, although now it's been peer reviewed, and this is the version of the article that will show up kind of as the for the record version of the article. It actually, so this isn't totally new. I'm kind of revisiting this. The preprint, which is a different title, but same group and very much the same data, some slight editing. This came out in September. And in fact, for our aficionados, some of you might have already heard a treatment of this on TWiEVO, episode 809, Vampire Vax, which I think covered that preprint at the time. And then also these vaccines, maybe for rabies, in bats, actually, to try to lower bats for spillover potential. But I really wanted to, even though this has been covered a little bit on TWiEVO, I really wanted Vincent and me to pick this up and kind of kick it around a little bit and discuss it, because I think there are some really important features in here, sort of getting to Priya's question a little bit about some themes about how coronaviruses and closely related coronaviruses, the sarbecoviruses, which includes SARS-1 and SARS-2, sort of how they, as you sample around, existing diversity, what you can infer about how these viruses, where they are, what they might do, how they might evolve. And then this might give us some clues for thinking about, as we're already talking about, sort of, not just sort of the next variant, but different serotypes, or maybe even new spillovers, potentially SARS-3, SARS-4, if we're years down the road here, hopefully many years down the road, decades or something like that. But certainly I think some important clues about the evolution of these that might be really useful to guide future surveillance, and also to think about sort of the whole mix of pandemic preparedness.
By the way, Nels, the last time we talked about this, someone from that country told me I was pronouncing it wrong. It's not Laos, apparently.
Yeah.
I think it's Los or Lowe's, something like that. I have not forgotten.
Yeah.
But Laos is not right.
Okay. Yeah. Thanks. Yeah. I mean, if I was
I think maybe
being
Lao,
graded on
maybe just Lao. Yeah.
Yeah, that sounds right. Yeah, if I were being graded for my pronunciation, I mean, I should be kicked off all of podcasting altogether. But yeah, no, a really good point. And speaking of that, so I wanted to, at least cover a few of the authors' names here, to lift up their work and who they are, which will also involve some mispronunciation. But so
let's
first,
bring it up here so we
yeah,
can see.
No, thank you. Yeah. So it's a pretty big group, multidisciplinary group. When we think about these field studies, but so first author, Sarah Temam, then Kamsing Vongphathiloth. I'm sure I'm all over the map on that one. I'm going to just actually concede for a few authors across. Pick up this paper though. So the preprint, or the accelerated published version available, open access currently. It should be easy to get access to this. And then if we look at some of the senior authors, some folks at the Pasteur Institute in Paris, so Felix Ray, Sylvie van der Werf, and Paul Bray, and then the last author, Mark Eloy. Thank you. Okay, very good. So equal opportunity mispronouncer of names here.
So Felix has been on TWiEVO, Felix Ray,
a
couple years ago.
And I think actually, Vincent, I think you introduced me to Felix at an ASV meeting a few years ago, maybe back in Minneapolis. That was a fun introduction. A big fan of his work over the years. One of the kind of preeminent virologists.
That's because I'm a hyper node, Nels.
That's right.
Sarah once called me that. Sarah in Colorado. What's her last name? Sarah Sawyer. She was at a meeting and she's standing next to me, and I said, "Why are you standing next to me?" She says, "Because you're a hyper node, Vincent."
Yeah, I have the same strategy. It's sort of just like a beehive of activity in virology. Just hang around Vincent and you'll meet some interesting people. That's a good idea. So, the big question here, a question that's captured a lot of attention, both scientists and, of course, in politics as well, is on the origins of SARS-2. Where did it come from? What are and the obvious place to look is for the most closely related viruses. And this kind of worked actually for SARS-1, right? So there was, there was enough really quick surveillance, and then just enough kind of pieces of the puzzle to make that connection of, an intermediate host in that case, and to kind of trace that. Similar with MERS. And so SARS-2 has actually been a little bit different. It's been a lot harder. And that, in a sense, might reflect a little bit of just the early kind of pandemic reactions that were unfolding in China. And then also maybe just some features of this virus that make it a little bit different. And we'll touch on that as the authors do, with I think at the very least a strong and plausible hypothesis about this the progenitor of SARS-2 emerging probably directly from bats, actually. So what was known before this work? So the sampling in Laos and in sort of south of China. There is a virus that has been touted a lot, rat G13, and that's not because it's from rats. It's another virus of bats. The I always say Rhinophilus, which is also mispronunciation. It's Rhinolophus
Right.
affinis. That genome, so that sequence of that virus was found in 2013, coming up on 10 years ago, and its genome is about 96% identical to SARS-2. And so, on the surface, that feels like, oh, wow, that's pretty close. But from a kind of virology standpoint, that's actually a really different virus. And in fact, it's not just sort of this even 96% across all 30,000 RNA letters, but there's areas of lower identity and areas of higher identity. And so it actually turns out to be quite, from an evolutionary standpoint, and given how fast coronaviruses, other viruses mutate, that actually is a somewhat of a distant relative. And so, so here they set out to do some more sampling and actually find a group of viruses, the Banal viruses, BANAL. And I'm not sure, actually, do you know, Vincent, how that naming scheme arose?
Oh, yeah, they did it. I don't know offhand what it is, but I'll look for it in the paper. They do have it.
Okay, yeah, I missed that detail. But anyway, so they found some viruses, and we'll kind of dive into the details there. But before that, there's a great sentence in the abstract, and I just want to linger on this for a few minutes. So the authors say that SARS-CoV-2 presents a mosaic genome. I'm now starting to throw out my pronunciation of normal English words, but to which different progenitors contribute. And so this idea of mosaicism, or a mosaic virus, it's got fragments from different related viruses. This to me is a key feature, a key conversation that I think gets a little bit put to the margin sometimes. We're so focused on, okay, what are the newest point mutations to the spike gene that are happening kind of in real time over the course of weeks or months in SARS-2 as we're stepping through the Greek alphabet, as we're stepping through the variants. And so I really want to highlight that, or underline that this notion of these coronaviruses being mosaic genomes, meaning that you have, if you just are starting to do sequence comparisons, you might go for the first 1,000 letters, and that matches one virus that's sort of known pretty well. And then you go the next 1,000, it matches a completely different virus pretty well in terms of the sequence identity. And so we'll unpack that. But I guess that's sort of my, putting the punchline first a little bit. I think that's my take home for this one, is to think about these viruses as mosaics. And actually, maybe it's a single genome fragment, or a single genome, or roughly 30,000 letters. But because this amount of recombination, this mosaicism is so high, in one sense, you can almost, it's not totally like influenza, but where it's separate segments, but you can kind of drop these segments in and out of the same single genome. And so that might have some clues about how this thing is evolving that I think we sometimes push into the background.
Now, as I think banal is coming from the sampling, like blood, anal swabs is part of it. That's what I remember from our discussion on TWiEVO, but there's nothing in the paper that actually explains it. I think it was in the preprint that.
That's a kind of an interesting thing too, actually. And so the preprint is also available publicly, so you can compare. Like if you're interested in the peer review process, this is kind of a fun way to sort of peel back that, or sort of look under the hood of that process. So I think the preprint arrived in September. Certainly they had submitted this as well to Nature at that time, and then you can see the date. So it then goes through the peer review process and ends up being accepted in early February, put out in this form. There's still kind of, the editors, the copy editors putting together the proofs, and they're doing that kind of level of checking for typos and other issues that might have arisen in the editing process, to then publish the sort of, for Nature, what will show up both on the website and in the magazine as well, which will probably be here in the next few weeks. But yeah, so to see, and in this case, my sense, I didn't do a really deep dive on it, but on the preprint, but the comparison is probably pretty similar. I don't think there are major differences here. How they kind of present some of the data, I think, are making some compromises for the form in nature, sort of the high density form, and putting a lot of stuff in the supplementals compared to in the preprint, but overall the same message here. And maybe actually to take a moment just to say this, that so, I think there was, and the reason this was on TWiEVO as well, there's definitely a big splash in the press about finding more viruses, of course, more closely related to SARS-2. But from I think the evolutionary virologist perspective, this was, maybe a splash, but it wasn't like a cannonball in the pool. This didn't sort of solve the origin questions. Where did SARS-2 come from? It certainly also didn't kind of quell the ongoing controversy about the idea of a natural spillover, which to me, I mean, I think what sort of circumstantial or indirect evidence we have still is just like a blinking light. It just makes, when we think about the virus dynamics, and we might, and thinking about these reservoirs and the sampling, we might revisit this as we discuss it, but all kinds of chances for a natural spillover. And then even some newer data about the early kind of cases in and around Wuhan, and how that really sort of congeals towards the market. But of course, this has been tied up in a lot of politics. There's a lot of people who are coming at this from different angles. And so that controversy goes on, and we're not going to solve that or break any new ground.
It's worth pointing out that these viruses tend to come from, as you mentioned earlier, the south of China and Southeast Asia, and they have in common a lot of these limestone caves, limestone karstic terrain in China, Laos, and Vietnam, and the bats live in those. Yeah. And so it's a common area for them to live in. As you'll see, they're not only they're similar species of bats, but similar viruses.
No, exactly. And so, yeah, let's dive into kind of the details here, because I think this is pretty remarkable, actually. So they captured a total of 645 bats. And by the way, worth it to read the sort of biosafety section of the preprint as well. This is, you don't just do this like on a weekend while you're picnicking. You really want to be careful for obvious reasons here. So 645 bats belonging to 6 families, 46 species. They got roughly 250 blood samples, 600 saliva samples, 540 anal or fecal samples, 157 urine swabs are collected, and this is in the north of Laos. And here, Vincent, I think you kind of like, it's easy to just go through the details here and not really take a minute to think about it. So 650 bats, 24 positive for coronavirus. And that might not sound like a lot, but that's actually a pretty small sample when we think about,
sure.
Right. And what is that, like roughly 5%, 2%? I'm not doing the math in my head properly here, but that's just kind of like on any given day. I think I was really struck by that.
Yeah, I mean, there are millions and millions of bats, right? So let's do let's get the percentage. The number of positives were 24, right?
Yeah, so 24
and they had 645. That is 3.7%. So let's say now 10 million bats. That would be 370,000 positives. So that's what happens when you get into the millions.
I agree. And even now, this is just the tip of the iceberg in a sense, because that's just on one day, one sampling.
Yeah.
And by the way, in some of these positive cases, they point out that actually there's certainly co-infections, and not only with like completely different classes of viruses, but even with coronaviruses, and again, in different, so like alpha and beta coronaviruses are coming out of one of the bats in just one swab. And so
they had one with both alpha and a beta coronavirus. And we should say they did a PCR, which would be a pan coronavirus PCR. So they're picking up many coronas. And so many were not positive, but many were. And there were 10 different species of bat, including the Rhinolophus individuals, but also others.
That's right. And so of those 24, then they narrow into 5 sequences of sarbecoviruses, and this is sort of the close family of SARS-related viruses, and a few others. And 3 of them are really close to SARS-2, actually, in some cases even closer than that rat G13. And so that's kind of the big deal here. And so now, 3 out of 600, I guess we're at like 0.5%, but we're at in that one sort of, endeavor of sampling, you've got 3 viruses that are closer relatives to SARS-2 than anything we've seen before. And as you're pointing out, Vincent, like when we start thinking about the millions or hundreds of millions, billions of bats out there, or hundreds of millions, certainly just in that location, those numbers start to really pop. And so anyway, so
they also point out 96% identity. So that's 4% difference. That's 1,200 bases in a 30,000 base genome. That's a lot.
That's I think quite a bit.
Yeah. I think Eddie Holmes has said it would take 20 years for that amount of change to be in a corona genome, just as it's mutating along from host to host, right?
Exactly. And so, and I've heard similar numbers. And so that kind of gives you the sense of, if you really want to trace where the kind of set of bats where SARS-2 emerged from, we're not that close. We're 20 to 40 years sort of away from finding that. And I think given these numbers, even in this small sampling, you can see what a massive task that would be. And I don't know, actually, I'm assuming there's a lot more sampling like this going on, but do you have a better sense of that, Vincent? Are there groups out there really hitting the bats hard these days, or what are you hearing?
I'm not hearing anything because a lot of this, this is independent of EcoHealth, remember, and EcoHealth was doing its own, but I haven't heard anything. I'm trying to get Peter Daszak back to talk about it, but it's not been easy. So I assume, like you do, that it's ongoing, right? Yeah. Since it's so important, right?
Agreed. Totally hope so. So looking and kind of getting to your point about the 96% identity. So going to figure 1 of the paper now in panel D, they do sort of a gene by gene heat map of how closely or what the percent identity is between these sequences. So you can look at, this. So perfect. That kind of bluish area there with those squares. So each of the exactly. Right, exactly. So each of those squares is a different gene. So, and not the same size. Yep.
Yeah. These are the different orphs, and there's the spike there at the bottom. Yeah.
Exactly. And SARS-2 is that top row. And then 2 of these these new viruses that they sequenced out, and in some cases domesticated. They actually were able to get the virus to replicate. We'll talk about that in a few minutes. But you can see, so that 2 of those hit higher than any or than rat TG or any of the pangolin viruses. And so here's an important part of those that already you can kind of just see by gazing at these squares is when things get light blue or orange, so that amino acid identity is going down.
Yeah.
And so already for some of these pangolins, even the bannels, that light blue in the
spike, right?
Spike, yep. The nucleotide or the N-terminal domain, I should say. That's a hint at this recombination. So it's like this sort of drop in identity and then back up as you go down farther in the genome. And that's one of these, this is a sense of that mosaicism. Even the pattern here starts to look like mosaic tiles, right? As you put together different parts of the genome and see these recombination events. And so, to say though, so the kind of big deal here, so rat TG is down, 13 is down the list. But that spike actually is sort of darker blue across the whole genome. And so that's why this one has been held up as, even though, as we're pointing out, it's like 1,200 differences, still like the closest in terms of the same set of fragments, roughly speaking. And then this.
But this Benel 2052 is rivaling RATG. It's mostly dark, right?
Correct. And in fact, it's more closely related. And so if we shift over to the left there in panel B, this is just sort of a more traditional phylogenetic tree.
Yeah.
And you can see those Banel sequences, the 236, 103, and 52 are all clustering there.
Now, so here's the virus up here, right?
Correct. So these are two
human isolates, right?
Yeah, and
very early ones. Previously, the RATG13 was the closest. But now you can see closer on this tree are these banals, in particular, not this one, but 2052, which is the one that scores really well there on
That's
the blue.
right. Exactly. And what's interesting is, so, and we'll go a little deeper on this. The way the sequence you pick to draw the tree will dictate how closely these things relate to each other. So if you took that 236 virus, that's shown sort of the fifth one down, and instead of, they're probably using the ORF 1A or ORF 1AB sequence for the treat. But if you use the spike instead, all of a sudden rat TG would come up higher, and 236 would fall down. 2052 would still stay higher or more closely associated or closely related, which was sort of the splash of this paper, is to find one that on the whole is more closely related. We also have that nice map there a little bit farther if you scroll.
Yeah, this is where it's coming from. Let's blow that up here, and you can see these are the sites where they collected the bats with the red stars, right?
Exactly.
So you can see, similar viruses have been picked up in Thailand. There was another study done before this where they found in a drainage pipe a little colony of 300 bats. Many of them had sarbecoviruses. So there's the border. And then, of course, we got China right up here. So this is all common ground for bats.
That's right. And four sites kind of distributed from north to south. But it also, maybe, I don't know, it's hard to illustrate, but just how limited the sampling is again. So it's 4 different sites,
sure.
But you could have literally millions of sites go across the border to Vietnam or to Myanmar. And there's, endless available bat caves for these things to be percolating around in. And so, we're just really barely scratching the surface.
All right. So thanks. Les is one of our moderators. He says banal comes from bat anal swabs. There you go. I was almost there.
Yeah, close. Yeah. And that's another, the alphabet soup of coronaviruses and people picking their own way of really tricky, but yeah, thank you, Les, for that clarification. So we have this whole set of viruses now, and what the authors do next is start to kind of put together a little more of a sophisticated view on how these viruses relate to each other. And so you've got the phylogenetic tree, but then they go ahead and start to do this recombination analysis, this breakpoint analysis. And this is what I was kind of, this idea of the virus being mosaic or composed of different fragments. This is what I really wanted us to sort of dig into. And so what they can do then is, they get these hundreds of sequences or pare down to a few dozen sequences, and then start comparing them and asking, is there evidence that some fragments don't match the others based on based on how, if you build a tree with different parts of the genome, how the viruses relate to each other. And if that initial pattern sort of breaks, and now 2 viruses that look very distant from each other all of a sudden look really identical, for example, in the spike gene, you infer that there's been a recombination event, and you would mark the breakpoints, as they're called. So it's like the genome is going along, a recombination event happened, it changes the identity really quickly, or really drastically in some cases, and then really quickly changes or breaks back to the original sequence. And so they use a program called GARD. This is a genomic algorithm or genetic algorithm for recombination detection. And then on a personal note, I was really happy. I was just scrolling through the methods last night and see that they use a newer tool called ID plot. And this was developed by Stephen Goldstein, who's a postdoc in my lab. And we teamed up with Aaron Quinlan, who's a spectacular genome scientist, computational biologist here at Utah. And so, I don't want to take too much credit here. So GARD, that algorithm, is really the main engine for doing the recombination detection. But what Stephen did with ID plot is just build sort of a more kind of user accessible pipeline so that you can do exactly this. So not only run the algorithm, but have a sort of a visualization across the genome, and then have it return the differences in the trees as part of just sort of a workflow. And so we're still trying to get that paper published. We have a preprint out, which is hopefully I'm guessing where they caught wind of it, but we're still working to get ours published. But so anyway, so using mostly GARD, but with kind of implemented in ID plot, they propose 14 breakpoints. And so basically 15 fragments from with the collection of viruses that they have here. And so remember, this is, they're aiming for viruses that are pretty closely, related. That goes out to the pangolins. So, maybe we could infer something like a common ancestor. I don't know, 100 years ago. This is just sort of a back of the envelope calculation. But within that time, you can almost immediately identify 14 breakpoints. And that really speaks to this mosaicism. So it's not, and this is different. In some cases, it maps to the genes themselves, the open reading frames. Certainly in the spike, which is a hotbed of finding or probably retaining recombination events. And so, and maybe to just step back. So, the idea here, right, is that the viruses are just doing this like a random mutation, the same way you might get a different letter. They're just, instead, it's sort of a different mechanism. They're just swapping in different parts of genomes as they're doing what's called template switching. So this is why those co-infections are, like, the fact that they're so easy to find speaks to how, a virus might be replicating. It's going along on the polymerase. There's another virus genome, even somewhat distantly related coronavirus in the mix. It hops onto that or switches onto that template, goes for a while, switches back, or there's sort of more events going on there. And so this happens a lot. And so, and in a lot of cases with pretty different viruses, pretty distantly related viruses, and that can kind of scramble the deck of the genome here. And so, I don't know, Vincent, do you want to throw up panel C really quickly, or even better?
C?
Yeah.
This one is the recombination analysis, right?
Yeah. Let's look at that. So, this kind of puts us a step ahead. So these are those 15 fragments that I was describing. Right. So we've got, and how it kind of maps onto the open reading frames. And so in the kind of red numbers or the red vertical lines, you can see the difference. It doesn't match perfectly with those horizontal arrows, which indicate the gene coding regions of the different open reading frames. But so then as you march through here, they could have used a different color scheme to really kind of drive home the mosaicism. But instead what they do is they have, and you have to kind of squint at it probably from the podcast, but it's worth, if you're looking at the preprint or the paper, worth taking a look here. So they basically, for each of these fragments, they label it for which is the most closely related virus sequence that matches the genome of the virus they're thinking about. And so SARS-2 is at the top, then they have rat G13, and then there are 3 bannels that they discovered and sequenced and analyzed here. And so as you go from fragment to fragment, you can see that the closest match, and this is basically from a blast search, where you're basically taking the sequence, in this case, the fragment from SARS-2, and then asking all of the virus sequences out there, coronavirus sequences out there, is there what's the highest identity? And so for that first fragment for SARS-2, it's a virus called RMYNO2, right, from a different species of Rhinophilus. No, Rhinolophus. Thank you. I'm never going to get that. And then the second segment is one of their viruses that they just pulled out, not one of those 3 but one of the other bannel viruses. And then in 4, they have this code multi. So there's several viruses that basically they can't distinguish which is the closely matched. Several that are a very close match. And then you go to fragment 5. It's a totally different virus from a different bat in the genus. And then multi different, the same as the first fragment. And then as we keep going, there's another bannel. Or I think are we past the spike yet?
Spike is at
here
10.
It is. Yep. Yeah. And so there we see rat TG13, which is an interesting, another reason why, even though it's not on the whole the most closely related, or was up until now, but the spike is actually really close, at least the first half, the N-terminal domain. A lot of viruses showing up in the S2, the receptor binding domain, which they spend a lot of time thinking about for obvious sort of functional reasons. And then we're back to multi, and different bannel, bat, and multi. So that's just going across one. And then now imagine doing that for the whole list. And a lot of them are just like SARS-2 is kind of borrowing, in a sense, all of these sequences from other related genomes. And then this thing pops up. And so I wanted to make this kind of pause here to make that bigger point that when we think about, okay, are we going to find the SARS-2? one of the difficulties here is, what does that mean even when it's borrowing from so many different pieces? And so certainly there could be a direct progenitor. And in a sense, instead of even thinking about what's the total amino acid identity, or the RNA or amino acid identity, if you do the translation, to think about what's the highest like recombination identity. Do those fragments match that sort of code of borrowing from those things to get at who really the closest relative is? A little bit of a different way of thinking about it.
Somewhere in this paper, they make the statement, which is, we'd like to find the immediate ancestor to SARS-CoV-2, but the best we're probably going to be able to do is find all the parts that went towards making it.
Yeah. And there's a lot of useful information in that, because, and now this gets also, I think, in some sense, we really do want to find the closest relative of SARS-2, or the closer we get to that, it could add some clues, at least indirect evidence or circumstantial evidence of sort of the chain of custody for spillover and some of those features. But in a bigger sense, I really think, and maybe as we're sort of hopefully catching our breath here in the SARS-2 pandemic, what are we doing? What are we learning about these viruses for the years ahead, for not just, this summer, but let's say a decade from now? And do we know what's sampling what, and what are the chances of these things sort of spilling over? And can we take some measures, even in some cases, to really say, for folks living in certain areas, like are they part of the science here? Are they understanding that how you deal or interact with these bats kind of could make a difference? And so, a lot to think about, I think, but also sort of that bigger view of learning about how coronaviruses work.
I think it's worth pointing out that none of the parts of SARS-CoV-2 is from a pangolin coronavirus sequence.
Yeah, that's a good point, Vincent. Yeah. And that was kind of, that was sort of the ripped from the headlines situation a bit earlier in the pandemic. I think that idea is kind of really cooled off, especially as these Banal sequences come online. That sort of is, and with some of the functional data they do, that we'll move on to now. That starts.
By the way, there's a good question. How does this mosaicism compare to SARS-1? I remember they never found the exact ancestral, but a cave with bats that had all the pieces. That's absolutely right. And they never found the ancestor, but they found a cave with the pieces. And I don't remember how many pieces there were. Do you remember, Nels?
I don't. Yeah, it's a great question. And Stephen, and so his ID plot preprint, I can't remember if he took a closer look at SARS. He took a look at SARS related, I think, from a little bit of a bigger picture. And so that's another, I should say it's a really good question. And it's also slightly arbitrary based on the comparison that you make. So the sequences that you pick will sort of start to give you, as you do those comparisons, it's sort of based on those trees with the dozen or 24 viruses, whatever. And so, if you pick one set of 24 viruses, you might have one set of fragments. If you pick another 24 in the sample, you'll still have roughly the same, but they might be slightly different fragments sort of based on that. And so I think it's a great point. It's a great question. And worth a look. So basically what you would do is take SARS-1 and, the equivalent of sort of the most closely related viruses to SARS-1 that you could find, and then build a similar, or do a similar analysis. And, my hunch is that it would actually be very similar, that you have this sort of level of recombination of mosaicism going on. Some of it actually doesn't, necessarily mean, and again, sort of the difference between, so it doesn't necessarily mean that, mosaicism doesn't equal danger. But so there's plenty of recombination events that like make the virus sort of worse or less able to potentially move into a human host. And it's sort of litigating this out in nature. And so a lot of it might be neutral. And then it's sort of the rare combination that we still don't understand, by the way. So we know a lot about, we're learning a lot about the receptor binding domain in particular. We're putting all kinds, for good reasons, putting all kinds of energy into the interaction between the spike and ACE2. But we know a lot less about, or just have some hints about how the combinations of different things might really sort of thread the needle, right, for a human spillover and infection.
I want to make that point because they say in the introduction here, the spike sequence is important, obviously, because it gets the virus in the cell. And then they say it is responsible for host range. So spike ACE2. It is not responsible.
No.
It is necessary but not sufficient, because once the virus gets in, other things have to be right? All the other proteins have to work in the cell. That's called permissivity. And I think this is not correct to put it all on getting in, right?
I 100% agree. And I think we've become, that's like, so the most is known about the spike. You can kind of, and I'm not trying to downplay it completely. Obviously, there's plenty, given that this is on the surface of the virus, there's plenty of epitopes that the antibodies can start to recognize. That interaction, of course, matters. We'll maybe touch on the furin cleavage site here in a few minutes too. But yeah, exactly, Vincent. And they bring in a tiny bit of this. For example, ORF8 is lost in many cases, right? So
That's right.
We've talked about this, and this happened in SARS-1 as well, that as the coronavirus moved from an animal reservoir into humans, give it a little bit of time, and usually in the context of infecting humans, ORF8 is lost. And so what's interesting about these banal viruses is they have ORF8. And as well as the obviously a SARS-2, and we've seen that lost in a lot of cases. But so that actually, so the fact that ORF8 is there argues that virus up until, or this kind of collection of viruses up until now, hasn't been circulating widely in humans. It's maybe been sort of nibbling at the edges or interacting, right? And so that's an important point that sometimes gets, again, pushed to the margins as we're thinking about, these fanciful and complex, in a lot of cases, complicated ideas of lab things. And it was, kind of nefarious things happening. But so far, at least, the indirect evidence is, continues to sort of just add the drumbeat of, nope, you can find a lot of these viruses in bats, and there's people interacting more and more with these bats. And so, okay. So anyway, so that gets at this mosaicism and sort of this different combination of fragments here. And then they do, and for good reasons, begin to focus on the receptor binding domain of the spike protein. And so they do a number of binding assays, both kind of ranging from in vitro to more closer to what things might look like in an infection. And they note that the Banal, in particular, this Banal 236, is more efficiently at binding the human ACE2. Again, sort of just like out of the box, right, in the wild, collected in 2019 or 2020 when they were doing the sampling, when they were doing the work, actually binds better to human ACE2 than the initial Wuhan 1 strain. So this is just like at the turn of 2019 into 2020, the very first sort of index cases, or as close, no, I don't think we have the index case, but as close to it as we've come, which defines the two original main lineages, A and B. And this guy binds better. But they do note there's no furin cleavage site. And this does remain a bit of a mystery. I'm curious what your current thoughts are about the furin cleavage situation, Vincent.
So these spikes from these Banals bind ACE2 pretty well. And that's in contrast to RATG13 spike, which doesn't bind ACE2 very well. That virus does not infect ACE2-bearing human cells. So what we see here is that just randomly out there among bats have evolved some sarbecoviruses that can bind ACE2 with reasonably high affinity. And you wonder where that came from, right? But that's another question. And they say basically we don't need to have any other intermediate host to get pretty good ACE2 binding, because they do show in a bit, you'll probably get to it, I don't know, that these spikes from these Banal viruses can bind ACE2 and allow pseudoviruses to get into cells. And in fact, they even isolate infectious virus and show that they infect ACE2 cells. So you don't need an intermediate host. You don't need pangolins. All right, the furin cleavage site. The bat viruses don't have this. They have no idea. Maybe they didn't sample enough, and maybe they are in bats, because we only have less than 100 bat sarbecoviruses probably.
That's right.
And so maybe it arose somewhere and we just missed it. So I think you can't really put too much on that at this point with so little data, basically.
I agree. No, I agree with you. And by the way, this could be, another fragment that comes in, another recombination event that we're just and I agree with you, Vincent. So we just haven't sampled enough here. But we also, so outside of, a couple of labs, Mark Dennison's lab at Vanderbilt, one or two others, we really don't know a lot about how kind of the features of recombination in coronaviruses.
Sure.
And so, right. And so we can, here the algorithm is calling 14 breakpoints or 15 different fragments, but, and these are different sized fragments. You can start to, like, if you tune the algorithm onto the spike where there's probably, at least a priori, there's probably just as much recombination activity, but natural selection is acting, because that's sort of the real game or a big part of the game here for maintaining host recognition and
right.
potential for spillover, et cetera. But there could very well be these recombination events that bring in furin cleavage sites. So I think that's part of the controversy. I have to confess, I haven't been following it that closely, but and again, from, I think, some sort of suspicious actors who have an agenda to push on where SARS-2 came from. But, how do you just sort of out of whole cloth get a furin cleavage site, which is sort of a semi-different, of several amino acids that just don't sort of show up very simply from point mutations? Well, you just drop it in as another fragment, I would propose, right? That there's a recombination event that could bring this in. And so, yeah, I mean, another reason to keep the sampling going is I wouldn't be surprised if we start to uncover pockets of coronaviruses that actually have furin cleavage sites hanging out. And certainly that wouldn't be completely new. There are furin cleavage sites weren't sort of totally new to SARS-2. This has been seen in coronaviruses. We have a question here that you're bringing up, Vincent.
Yeah, what about copy choice recombination for furin cleavage site? I mean, yeah, that's what Nels is talking about, basically, a recombination event. Close. I mean, there are clearly furin cleavage sites in other coronaviruses. Yeah. And even in bat coronaviruses, there's something close, but not exactly right. So, I mean, we basically undersampled so much that you cannot say it's not there, right? That's the bottom line.
Yeah, absolutely. Yep. Yeah. And there's several sort of flavors of recombination, as our listeners pointing out, that could get you there as well. I mean, the other thing that's kind of wild here, I think, is so thinking about the receptor binding domain, remember, like, these bats I forget how many species, it was almost 50 species of bats that they were sampling in this collection.
In this particular study. Yeah.
Yeah. And so, and with that identity to ACE2. So one of the really interesting things about
46 species. Yeah, you got it.
So one of the really interesting things about, the evolution of ACE2 actually is that it's pretty, interesting in bats. There's these signatures of rapid evolution that we talk about a lot on TWiEVO, which, by the way, is missing in the primates. So, of course, we have ACE2, but if you take the sequence of ACE2 from humans, from gorillas, from macaques, from neural monkeys, it's actually, it's pretty similar, almost identical. Whereas as you sample a collection of bats, you see a lot of differences in ACE2. That would be, I think, a fun future direction, if it isn't already happening with this work, is to sort of ask, we have these viruses, and you can very quickly do some of these in vitro assays. Where did the virus come from in terms of the species? And are there any sort of clues about recognition of ACE2 differences among closely related bats that somehow maybe maps over to the more stable human situation? Is there sort of just this coincidence that, given the diversity in ACE2 in bats, that it sort of predisposes some of these viruses to recognize humans kind of right out of the box? And I would be surprised if people aren't doing that. But as more sampling is happening, or trying to correlate this to the species they're from, there could be, I think, some interesting sort of evolutionary signal there to think about.
I mean, I also think we could take a clue from the evolution that we're seeing in the pandemic. I mean, the receptor binding domain is changing to allow, in many cases, better binding. And, you can imagine that happened early on when it first spilled over into people, right? There was a period where maybe the spike wasn't optimal, but it got slightly better. It was good enough, like maybe these viruses, and it got a little better on passage. It's totally within what we see now, right?
Absolutely correct. And that, it points to all of we really worry about laboratories, and we should. So BSL-3, BSL-4 work, but we're doing the viruses, the bats, and us humans are doing this at BSL-0 every day in these interactions. And so of course there's going to be all of this sort of proximal evolution that's happening as this is before this really, unfortunately spilled over and then really became a pandemic. So now we've kind of stepped through. So the authors have done this sampling, a bunch of different swabs. They've done a lot of the computational analysis, including, to point this out, including the binding assays. And then finally, we get a little bit into the virology here. And so the first step they take is to pseudotype lentiviruses with the spikes from Banal 236 in order to compare that to Wuhan 1, the index case or index-ish case of SARS-2, and then infect 293 T cells plus or minus expressing the human ACE2 receptor or not expressing the receptor. And what they see is just as we've said, so that actually the 236 is the pseudotype viruses are doing fine in the 293 Ts. In fact, I think, is it true that it's more so than Wuhan 1, or is it's roughly the same, if I'm remembering right?
Yeah, it's roughly the same. Yeah.
And then, but this is pseudotype virus. So this is, again, that focus, the very reasonable focus on the spike protein. But then what they do next, I think, is what really sort of starts to elevate the paper a little bit, which is to actually to kind of semi-domesticate the virus, right? So they've got all of these swab samples. And I think they took one of the bat rectal swabs of this 236. So they had several samples of that bat where they got it. And then they put that sample onto viro cells. These are old world monkey cells, and actually get small plaques. So here's like some real virology. So just enough of the virus is able to replicate. And the reason they're using viros is just this has kind of become a normal thing in coronavirus biology. It's a very permissive cell for diverse coronaviruses. And then using kind of that foothold of those small plaques, which means, by the way, the virus isn't doing that well. It's just sort of inching along. But they can collect some of those virus particles and then put it on human cells, the CACO2 and the CALU3. These are airway epithelial cells, if I'm remembering correctly.
CALU or airway, CACO or colon epithelial.
I see. Okay. So both epithelial from different locations.
Yep.
And they actually observe, I have to say it's modest, but they observe modest replication in human cells. And so, that's different than infecting a human itself through a complicated transmission route, but it's still it's pretty remarkable, right? So you started with 600 started with 600 bats. You found way more coronaviruses than I think you might have expected just from dipping the sort of shooting fish in a barrel almost. We're kind of this fishing expedition pulling all these out, 5 of which are sarbecoviruses in the family, 3 of which are more closely related to SARS-2, or at least in some parts of the genome, than we've seen so far. And so
that's what we were excited about on TWiEVO, that they actually had a bat isolate related to SARS-CoV-2, which is infectious, right? So this has been out 236. This is the first one. For RTG13, we don't have infectious virus for that. So this is the first one. This is quite close. But again, it's still 1,200 bases different from SARS-CoV-2. And for those people who are thinking, somebody had a closely related virus in the lab, no. This is the closest they've ever gotten, and it's still not close enough to be ancestral to SARS-CoV-2, right?
That's right. Exactly right. So there's still a pretty big gap here. So
Nels, they say, these data support the hypothesis that SARS-CoV-2 could have arisen by a recombination event of sequences, note they say sequences, pre-existing in Rhinolophus bats living in the extensive limestone cave systems of Southeast Asia and South China. And so they say, people who collect guano, or people who spend time in caves, tourists even, are at risk. And so that could have started this. Who knows, right?
Absolutely right. Yep. And that's, it's not certainly not a smoking gun or like direct, here's the one-to-one connection, but it's the closest we've seen so far. And
Yeah.
and I think, and that's the trick here too, right? Is the sort of how do we communicate the uncertainty here, or connecting those dots, that those extra dots, there's nothing sort of so suspicious or nefarious about that. It's
Yeah.
just sort of the problem that we're up against. It's sort of the biology where we find ourselves in the amount of resources that we've put into it so
No,
far.
this is a needle in a haystack, literally, because there's so many bats, as we've said many times, 20% of mammals, and there's millions and millions and millions, and we've barely sampled them. So to really know where it came from, and we don't know. I mean, Southeast Asia is a big geographic area as well. So we'd be lucky, considering the small amount of effort we put into sampling. And you can see here, this is a big effort, and it's still nothing.
No, that's exactly right. And then it's, you take the millions of bats, but then what do viruses do? They replicate even more. So, for each of those millions of bats, think about potentially billions or trillions of virus particles, depending on how it's sort of cooking through there as well.
Sure.
And it's, I don't know, it's this, I've been kind of playing around with these ideas about how most of virology is invisible, which is sort of obvious in one sense. But I think we even when we think about it, we underestimate it. Our brains aren't like sort of tuned to like understand the magnitude of this and what it really means in terms of the just sort of daily insults that sampling those natural experiments that the viruses like this are doing every day.
By the way, I just want to point out, we're impressed by bats, but rodents are 40% of all mammals, so even greater. And they live in proximity to more humans because we have rodents here in New York City and many other cities.
No, absolutely right. Yeah, so I'm glad you brought up that point. So, and that was in Stephen's preprint, introducing ID plot. He took a deeper dive on a different set of coronaviruses, the SADS related coronaviruses. These, and I'm probably not remembering the exact acronym
Severe
right now.
Acute Diarrhea Syndrome coronavirus.
Yep, exactly.
SADS. Yeah.
Yep. And so there you see both not only activity in rodents, but also in livestock. And so, when we think about factory farming or even just traditional farming and those interfaces between rodents and livestock, pigs and cows, and by the way, the patterns of recombination are very similar. You see, if anything, you might even see some of the comparisons that Stephen did, even in a very small number of virus samples, like 8 or 10, a dozen. You see 15 of these recombination fragments or these mosaic genomes. And so, there's that kind of virological conversation happening, not just in the bats, but also in livestock and rodents, as you're pointing out. And so
SADS is a bat spillover into pigs that happened in China and caused a huge outbreak in China. They had to do a lot of culling, and the pig industry was affected. So these spillovers don't just go into people; they can go into others as well.
Yep. And then and with the potential of these emerging from livestock. So I think, OC43 back in the 50s, our best idea is that came from cows to humans, which now causes the common cold, one flavor of the common cold.
Which you can remember because the name OC could mean old cow. It doesn't, but you could...
Yeah, this is the good thing about alphabet soup. You can kind of tune it for your own memory system there. No, so anyway, so that's the paper. And just as it was coming forward, when we were back in September, I really wanted to cover it. I knew it was happening on TWiEVO, and then Vincent and I were both sort of scrambling on multiple fronts. Our podcasting sort of, we're back and running strong in 2022, but we had a little hiccup there.
I always want to hear your opinion of these things too, Nels. So when I saw it, I knew we had done it, but I wanted to do it again.
Yeah, no, thank you. It's really fun to
Should we take a couple of questions here before we?
Oh, absolutely. Picks? Yeah, great idea. Yep.
So Andrew says, would understanding the mechanics of the mosaic gene patterns make it easier to predict possible future evolution?
Yes, this is a great question, Andrew. And this notion of prediction with viruses, I don't want to go too far out on a limb here and say that we can like cleanly get the rule book for saying, here's SARS-3, here's SARS-4. I don't think it's quite that simple. And if anything, these viruses have, a lot of surprises in store for us. As they're these kind of master evolutionists. Maybe I can't remember exactly what Vincent said at the beginning, but I do think it's important that we understand I think the authors rightly kind of lift this up as a concept, like of thinking about SARS-2 as a mosaic, of thinking about coronavirus as a mosaic, because what it does is it sort of shifts our thinking a little bit away from, I think, sort of that, what happened last week in terms of 4 more mutations in the spike, to you can kind of drop in and out big, fragments here of different sizes. And it's sort of a different tempo of evolution or a different style of evolution. For me, what it underlines is that we really would do well to understand how these fragments or these parts work together as it relates to humans or other, bats or pigs or cows, because then we'll have a better understanding of, maybe not predicting, next, 3 years from now, this virus is going to come from here, but that here's a set of viruses and the patterns, those are, maybe it's 1 or 2 fragments away from really being what looks like a virus that's efficient in humans. And that in the end, it might not be. There's kind of a lot of secret hidden virology here in how coronavirus is working. We're kind of in our infancy of understanding these viruses at that level. And so, but I do think, so that's where I'm coming from in terms of, not predicting like a thumbtack on the map or something like that, but really kind of the classic foundational sort of studies in virology that have really taught us enough to kind of make it through to do better. So it's hard to do worse, to be honest, like with where we are. I think we've put in tremendous resources into our kind of daily surveillance. And in some places, and my God, I mean, the vaccine rollout and the timing of that is, a real breakthrough. But I am a little bit worried that with the hunger for this more foundational science, is hopefully it's building. It takes longer, but I'm really hoping that there are new labs and that sort of emerge and take this on as their people's careers. I think there are many fruitful careers in coronavirus biology that will hopefully echo on. But that depends on foundational basic science being funded and people sort of going into the profession with some of the headwinds out there.
So Kristen wants to know, is the full genome sampling of any virus equal to SARS-CoV-2? Yeah,
great. Oh, in terms of, I can't think of anything. I mean, the closest maybe we come is influenza. And in fact, it's a great question. It's interesting when we see how sort of genomic epidemiology pivoted into SARS-2 as that emerged. A lot of the groups that had sort of the infrastructure in place or the tools or related resources in place were folks that were thinking about seasonal flu. And so we don't have that concentration or the number of cases every year the same way, like with this sort of burst of sampling that we've done of SARS-CoV-2. But over the decades, it starts to, it's the closest we can come. It's still, like, I would say at least an order of magnitude lower than SARS-2. Yeah, it's pretty wild how everyone kind of pivoted into this, and the number of sequences that have been sampled and continue to be. Yeah.
So Jamie wants to know, why don't we have more viruses emerging from the Amazon? So there are already quite a few. You just don't hear about them.
Yep.
Right. And they don't end up causing global pandemics, but they do cause a number of problems. I have a slide in my virology course, by the way. So just go on this YouTube channel. You can find Virology Live. I think it's the Emergence lecture, which is down towards the end. I have a map of the Amazon North Basin and all the viruses that have been identified, which is just a fraction of what's out there. So I just think that doesn't get any press.
Yeah. Well, and then there was, I'm already forgetting the specific variant. I think it was before we were into the Greek letters, but there was a variant that really emerged and swept in South America, in Brazil in particular.
Yeah, the beta, I think it was, right?
Yeah,
P1, they called it P1.
Exactly, it was P1, and then maybe it was given a Greek letter, and there were people sounding the alarm in South America, right? Because I mean, it was one of the earlier waves with a really big impact on that population. But interestingly, this is kind of right before Delta came in and really kind of sopped up all of the oxygen, and that kind of it didn't emerge, it didn't compete outside of South America in a way. And so, and that's I don't want to get too far down the road on this idea, but there are definitely differences, regional differences in human variation and how viruses sort of sample their hosts and then and change. That's another area where we're really kind of in the dark about how those sort of factors influence that. But I wouldn't count out any region on the globe as not having the potential for a spillover into the future.
I want to thank Sandy for your contribution to our work. Much appreciated, Sandy. And also Priya. Thank you very much.
Fantastic. Yeah.
And also Buzz. Thanks all. Great incubation in progress. Thank you very much.
Very cool. Thank you all. Yeah.
What is the chance we get a spillback from the many mammals that get infected?
Oh, yeah. This is thank you for this question too, Jamie. So this is really, I think, an interesting area. And I have to confess, I was a little bit skeptical maybe even a few months ago, but I'm becoming more interested in that question. And I'll certainly let Vincent weigh in, who I think has been tracking this a lot more closely than me. But this is for real, right? So some of the sampling, like not at the level of so if you have 600 samples and getting 24 back of just sort of broad viruses, but there's some sampling where you're giving more returns of SARS-2 from deer, from other. So obviously the mink situation and how that's been leading to the culling of a lot of mink farms, including here in Utah. But my impression so far is that outside of the hypothesis that we entertained on TWiEVO a few episodes back about the origins of Omicron, and could it perhaps be one of these that goes into mice and then spills back. So it's a plausible hypothesis. We really dug into that in a paper that tries to make the case based on sort of the types of mutations that are seen in Omicron relative to some of the other variants. And some combo of immunocompromised versus spillback kind of things. But I still have to say I haven't been totally convinced of spillback. It seems like the viruses are hanging out in the deer and kind of doing their own thing. I even saw, I think maybe a new report that the alpha variant is still sort of having its moment in the sun in the deer population. So it isn't. So there's a spillover there. An initial one, and you'd imagine that there have been just as many opportunities or more so for Delta, for Omicron, but that Alpha is still sort of dominating in the deer populations. And so, that speaks to that it's not sort of a one-for-one relationship, that there can be a really different sort of virology that happens in a different host. Yeah.
So Aerozone wants to know if mosaicism accounts for the creation of SARS-CoV-2. Isn't it likely that recombination is ongoing in some unknown number of bat colonies?
Yeah, I that's exactly right. I think the short answer is we don't 100% know that, but it's I think that's kind of maybe where Vincent and I are pointing today is or calling for, right, is not only what's going on in the spike, which is certainly part of the story, but how do those other pieces of the genome, the other open reading frames, work together in a way so that once you get into a human host cell, that the virus can replicate somewhat efficiently. And now this is all of those other genes. So ones that are encoding proteins that are inhibiting immune functions, keeping the immune system at bay so that newly infected cell doesn't immediately get crushed by the host immune system. And not just on the antibody side. So the hundreds, maybe the thousands of innate immune sort of responses that our immune system is an incredible immune system that we all carry around with us. And these viruses it's not an easy job. It's not just sort of find the host cell and avoid the antibodies. There's a whole nother collection. The viruses themselves, right, have to go through the mechanics in a host cell where they can't bring everything they can't bring all of the tools they need to replicate. And so by necessity, they have to access some of that host machinery and use it to replicate. And that host machinery, by the way, looks a little different in a deer than it does in a human, than it does in a bat. And so now, and then as you put that together in a collection of, 30 or so genes total, or give or take, there's a lot going on there, and we don't understand how that works together. That's a really complicated question.
Richard, thank you for your contribution. Spike myopia needs glasses. Great to hear further explanation of concentration, co-infection, recombination. I first heard about this from Ralph Barrick, whom I discovered thanks to TWiEVO. We're trying to make scientists better known, right, through this podcasting effort, right, Nels?
Yeah, absolutely. And Ralph's a great one, by the way, working on coronaviruses for a long time. All kinds of great labs that have spun out from the Barrick lab as well, who are on the scientific front lines of trying to get a better handle on coronavirus biology.
And thank you for your support.
Thank you very much.
Of the incubator. And we welcome our second moderator, Tom. Thank you for coming.
Fantastic.
Let's
Appreciate
see
here.
Tom. When is the next TWiP out? It's in the can. I have to edit it. So I edited 2 TWiVs and an immune this weekend. I cannot do it today because I have to go teach after this.
Yep.
But I'm hoping to get to it tonight and tomorrow. So in the next couple of days. But thanks for asking. Really appreciate it. All right. So Priya says the cleavage sites usually need temperus at the sign of attachment. Okay. Yeah, that's true for, but the virus cells that Nels mentioned, they don't have surface temperus. The virus actually gets endocytosed, and there's a protease in the endosome that cleaves the spike and leads to fusion. But in the respiratory epithelium, there's surface temperus. And yes, there is camostat mesylate, which is an inhibitor. But given the brouhaha with hydroxychloroquine, I don't know if that's ever going to go anywhere.
Yeah, agree.
It's a real problem. Let's see here. Yes, unlike first SARS, this one spreads asymptomatically. So that's a big problem. Why it's done so well, right?
Yeah, agree. And another sort of example of this invisibility of viruses, the asymptomatic infection. That's something my lab we're putting some real energy into is that exact point.
Have any SARS-2 variants circulating in humans shown this mosaicism? I think we've talked about that, right, Nels?
Yeah, we have. And so the answer is yes. And so, so not surprisingly, there have been good evidence of co-infections in humans. And so in these cases, given just the dominance of SARS-2, we're starting to see cases where, or at least limited evidence, where really highly related SARS-2, in maybe more rare cases, like someone might be co-infected with 2 variants at once, but a little bit of evidence that there's recombination happening there. And so this is using similar phylogenetic analysis, the GUARD tool, and even just sort of some custom, like going by eye. So looking at that proposed breakpoint, asking what are the number of mutations so the difficulty here is to distinguish between, is this a real recombination event, or did the virus just sample the same, dozen mutations in this region that are different or part of this other variant, for example. And so you don't have an airtight case, but you build an argument on parsimony. What's the simplest explanation? And it's a lot harder to hit the 14 exact point mutations versus one event where you just drop in that fragment that has those 14 changes already. And so there's some good evidence from Andrew Rambeau's group and 1 or 2 others that, yes, this has been happening as the SARS-2 pandemic's unfolding. And then, of course, you get some of the news headlines that are a little bit over the top with the Deltacron variant and all of this stuff. So the answer is yes. And at the same time, no, don't worry about the notion that there's going to be some super SARS-2 because of this. Is just the coronavirus is doing what coronaviruses do.
By the way, Nels, Daniel Griffin likes to say that Occam was not a physician.
Yeah, that's right. Don't chase. What's the other sort of famous saying in the medical circle? Don't chase the zebras.
If you hear hoofbeats, think horses, not zebras.
Not zebras.
Right?
Yeah, exactly.
Don't think of the unusual thing.
Yeah.
Have you noticed an uptick in classes? I've taught a lot of virology, so I teach every spring at Columbia, and this year, right now, I'm teaching. Have the usual enrollment, but I have also taught 2 summers since the pandemic started. I never did that before, but there was enough demand such that I had that. So I'm going to be doing that for some time. So yes, there is.
Yeah, great question, Kurt. And
You don't
I
teach virology, do you, Nels?
not at like a full class. I'll do some cameo lectures here and there. I actually do a class. I've done a class the last few years out at the University of Maryland with boy, why am I blanking on his name? Bill.
Bill
Oh boy.
Jackson.
Yeah, Bill Jackson. He's invited me to his class, and I do a few things here too. But I think actually this is a potentially a really good outcome. Is the more biologists thinking about viruses not only has like some really good practical implications for understanding viruses better, but viruses have been with us the whole way. A lot of our biology unfolds because
Indeed.
of viruses, and
Indeed.
so this unlocks the possibility of a new understanding. And so yeah, maybe my dream here, Vincent, is sort of sort of an echo of like how we saw soccer become more popular in the US, where
Yeah.
football and basketball was dominating the scene. Maybe my science analogy here is like in a playbook of neuroscience, et cetera, that more talented folks doing virology is like, I can't think of a better thing for science, actually.
Although YWSL says, just want to say, I don't need COVID to be interested in virology. I like nature science. So there's some people out there, I agree. But
Yeah,
I think a vast amount of interest has been driven by the pandemic, for sure.
no question. Yep.
Is there speculation about why bats specifically in Southeast Asia are so primed for spillover as opposed to, say, bats in Texas?
This is a great question. I don't know. So I'm curious what Vincent thinks, but I'll do a quick one, which I don't know. It's interesting though, so there are infectious microbes matter for bats in North America. And the most sort of daunting one, not for humans, but for bats currently, is white nose syndrome, which is actually leading to, extinction level events in a lot of North American bats. Maybe some hints that there is some resistance, some genetic based resistance showing up in some of these populations, which might sort of like at the last minute sort of save some of these species. But there's also, of course, other pressure on habitat, et cetera. So I'm not trying to say that like if you're getting crushed by, in this case, a fungal pathogen, white nose, that sort of precludes it from also being a spillover potential for viruses and other things like that. But there are some just maybe like some contingencies. If you catch a population at a certain blink of time, there's just a set of infectious microbes or pathogens that are hitting one population in one geography, or more concentrated in one geography, more so than the others. And so come back in 10,000 years and there could be, a set of viruses in North America that are doing the same thing, in South America that are doing the same thing. It just happens that this is the sort of the current of virological events is a match there.
Yeah, I think that's. I also think that the species are different, and that coronas are species. They're not species specific, but certain coronaviruses require certain, not just the receptor, but the intracellular milieu, and that's the bats. And if we really knew how they evolved over millennia, maybe that would tell us, right? But we don't.
We don't. Yep.
Tom says you convey a sense of intellectual excitement. That's absolutely true. That's why I asked Nels years ago to do a podcast, and I'm very lucky that he said yes. Not everyone gets it, and Nels is a busy guy, right? He's just as busy as anybody else, and he said yes, and for that I'm very grateful, Nels. So
I'm grateful for the opportunity too, and thanks for the kind observation, Tom. That's generous of you to say. It's hard not to be excited about science. I'll be honest though, so growing up as a scientist, I'm kind of a shy person. And so, and taking these sort of complicated scientific ideas, especially earlier in my training, before there were gray hairs in my beard, it would really be hard for me to feel like I could competently communicate science. It still feels hard, but maybe I'm just old enough not to care as much or something like that. But even more so than that, this experience, the chance to kick around science with Vincent, with other of our peer podcasters, Kathy, Brianne, Rich, the whole group, at a time where I think, as scientists, my pick of the week will go in this direction in a few minutes. This is a time for scientists to speak up on all topics, on climate, on viruses, on go down the list. And science matters now. Science has always mattered, but I think scientists speaking up. And so this is a small attempt at that, expression at that. And so anyway, thank you, Tom. That's a really kind comment.
I've known Nels many years, and it's a smart guy, and he can express himself, and also a nice guy. So it was a natural for podcasts. So it was in a bar that I said, "Hey, we should do a podcast." Right?
I remember it well, up in London, Ontario.
All right. Thank you, D'Alessandro, for your contribution. We're a great team. Yes, we are a great team. I really enjoy. This is the only, I guess Q&A is the other one-on-one podcast.
With the live streams,
with Amy. But this is the podcast that one-on-one, and I enjoy it very much. Although, we do have a lot of guests here on TWiEVO.
Yeah, and more ahead. I'm working on some scheduling now. I think we'll have some really fun conversations in the months ahead.
Do you know any good mycologists? I do. I would love to do some kind of mycology podcast, but it depends on finding the right person. Now that I'm at the incubator, I'm here most of the time. I can do more. I'd love to do that, and maybe I'll pick Nels's brain. Maybe he knows some.
Yeah, possibly this week in fungi.
Yeah, TWiEVO. All right, Nels, you want to do some picks?
Yeah, let's do that. So my pick of the week is an interview with Eric Jarvis at Rockefeller. Eric's an evolutionary biologist. He works on the evolution of,
That's
works
him.
on the evolution. That's Eric. Works on the evolution of bird songs, which is a really fascinating topic. And so this interview is less about that, but it's more about scientists and public engagement, actually. And, Eric shares his experience too, as African-American in evolutionary science and sort of molecular science as well. And how this sort of echoes on from, if you haven't seen it already, I recommend, we didn't do a live stream, but we did an interview on TWiEVO 74 with Joe Graves on that topic. And so Eric starts here with kind of that idea that we were just kicking around, that scientists, it's time to be vocal, it's time to speak up. But also, in part of that work and in his experience is, how do we bring more people into science? How do we bring African-Americans into science? And in evolutionary biology in particular. So we discussed this with Joe, both his path on TWiEVO 74, but also sort of the current state of representation and how out of balance things are. And so this sort of brings another example to light and sort of, I think, highlights that there's a lot of work ahead in science communication, sort of along the lines we've been talking about understanding a virus, a pandemic, a spillover, understanding any modern scientific topic and the complexity, bringing that and I should say, public engagement. That's not just the public includes other scientists working on other things. And so as we extend those conversations as well, that's sort of like all hands on deck for science, both lay science, professional science, and professional science kind of done in different flavors. So anyway, Eric has, I think, a really effective take on this, and that's my pick of the week. Is this?
I like this quote here. I used to tell my students the most important thing is resilience, but I no longer say that. The amount of resilience I had to have to survive should not be needed. That's true, right?
That's exactly true, and that's what I mean. I think to hint at some of that work ahead. How about you, Vincent? What's your pick of the week?
So my pick is an article in Reuters. There's been a new case of polio in the last year or so. Yeah. Malawi detects polio, first wild case in Africa in over 5 years, which is a wonderful achievement that you went 5 years without a case of polio. But now there is a case. And what it is a type 1 polio virus imported from Pakistan. Type 1 is the last of the 3 poliovirus serotypes to be eradicated. There's type 1, 2, and 3. 2 and 3 are gone. At least wild circulating 2 and 3. There's still vaccine strains. But type 1 is still circulating in Pakistan and Afghanistan because we can't get in to immunize and get rid of it due to conflict in the area. So it's always being exported to other parts of the world. And as long as you're vaccinated, you're okay. But if you let vaccination levels drop below, I would say 85%, then you're going to have cases. And that's what happened here. This was a young child. It was last year, actually, that this case, a 3-year-old child, got paralyzed in November of last year.
Wow.
And was not vaccinated. And so this is the problem with not vaccinating against polio, right? You have to keep it up because until we get rid of circulating wild virus, we're going to have this all the time. So,
and it's so close. I mean, hasn't it just been on the edge of eradication, but then it kind of
Yes.
keeps filtering back. Yeah.
The problem is that we can't get rid of it in Pakistan and Afghanistan. And that is because we cannot immunize. If we could eradicate, because we've done it everywhere else. So it's a good pointer. This happens every so often. There's a case here and there in different couple of years ago in the Philippines. There were a few cases. Always when you let polio vaccination levels drop, it's a big problem. And until the wild types are gone, you have to keep it up. You can't say, there's no more polio in my country, I don't need to vaccinate, because we're not countries, we're one world. It's all of us together, right?
Yep. No, and great lessons there, not only for polio, of course, but for measles, go down the list, for SARS-2, and what the global situation looks like compared to the more wealthy countries. Night and day difference in some cases.
Yeah, indeed.
Yep.
All right, that'll do it for another TWiEVO. We are very happy to have all of you here today, all 169 of you. Hey, we have 95 thumbs up. Get us over 100, folks. Come on, just push the thumbs up buttons down below the video window there. And that's so that more people can find out about us. Our goal is to teach science. That's it, nothing more.
Yeah, and expand the conversation. And that, I think I have to say, the live stream is perfect for this. These questions, this is so fun.
They're very good, aren't
comments. They? Yeah, I can't get enough of that. And I'm looking forward to the next one.
The live stream, and these are many people who were here last time, and they have good questions. So
fantastic.
Someone said mixologist. No, we meant mycologist, not mixologist. We know how to mix drinks right now.
That's right.
Well, actually, Nels is a beer guy, so.
True. Guilty as charged.
That's good. Yep. All right. You can find the show notes for TWiEVO at microbe.tv/TWiEVO. You can send us questions or comments to TWiEVO at microbe.tv. If you have a pick of the week, you can join us as well, or you can just come here to the live stream and ask them. And if you like what we do, we appreciate your contributions here on the live stream. But if you'd like to contribute otherwise, you can go to microbe.tv/contribute. We are a nonprofit, so your contributions are tax deductible here in the U.S. Nels Eldey is at cellvolution.org. That's his website. And on Twitter, he's L Earlybird. Great to see you again, Nels.
Great to see you, Vincent. And plenty of non-SARS-2 papers just hiding in the decks. So we're hoping to diversify the portfolio here, but we're also, as we mentioned, kind of in that group of virologists sort of holding our breath, wondering what's next, what's the next chapter for SARS-2, and we'll cover it in the months ahead. So thank you, Vincent. Great to be with you.
I'm Vincent Racaniello. You can find me at virology.ws. Music on TWiEVO is by Trampled by Turtles. You can find their work at trampledbyturtles.com. You've been listening to This Week in Evolution, the podcast on the biology behind what makes us tick. Thanks for joining us. We'll be back next month. Till then, stay curious.